Hello, everyone, and welcome back to another round of Human Physiology lecture. We are going to finish up our unit on the cardiovascular system with Chapter 20, which is going to cover concepts associated with blood flow, blood pressure, and circulation of blood through the blood vessels. So this first section is going to focus a little bit on the characteristics of the different kinds of blood vessels: arteries, veins, arterioles, venules, and capillaries. So without any further ado, let's go ahead and get started. So just as a reminder, we talked about this in Chapter 19 just because even though Chapter 19 focused specifically on the heart, we needed to have a good idea of what arteries and veins do. So you'll recall that arteries will always be conducting blood away from the heart, and usually it's going to be oxygen-rich blood, and veins are going to be conducting blood back to the heart, and usually that blood will be oxygen-poor. The capillaries that exist in between the arteries and the veins are going to act as the sites of gas exchange, nutrient delivery, and waste pickup, as we've discussed before. And we will talk more about that sort of concept not only at the end of this chapter, but we'll be talking a lot about gas exchange when we get to Chapter 22. So when I say that arteries usually carry oxygen-rich blood and veins usually carry oxygen-poor blood, you just want to make sure that you remember the two exceptions to those quote-unquote rules above. All right, let's talk about the composition of the different kinds of blood vessels. Blood vessels can have up to 3 different layers or tunics to them. Each tunic is going to serve a slightly different purpose in terms of the maintenance of the vessels themselves and how they promote blood flow through the circulatory system. The first layer or tunic is called the tunica externa. So this is going to consist of a fibrous layer of connective tissue that coats the outside of the vessel, and in a lot of cases this is going to serve to anchor the blood vessel down in place to make sure that it can't squirm or squirt around. So it really makes sure that the vessels become a part of the tissue or organ that is being supplied with blood. The middlemost layer is called the tunica media. This is the middle layer that is mostly going to be composed of vascular smooth muscle. So in the past, when we've talked about vasoconstriction and vasodilation as a means of regulating blood flow to different tissues, the tunica media of blood vessels is the target for those sympathetic signals, whether it's norepinephrine or epinephrine. And finally, the tunica intima, which is sometimes also called the tunica interna. This is the innermost layer of the blood vessel, and this is literally just a layer of endothelial cells of the squamous variety. These basically form a hollow tube, and they actually form the tube-like structure through which blood is going to flow. Now, what is interesting here is that while blood vessels can contain all 3 of the tunics that you see here, not all of them do, but ultimately every kind of blood vessel at minimum has to contain a tunica intima. You will see that some vessels do not contain tunica media, and because of that, they are not subject to vasodilation and vasoconstriction, and then some will not contain tunica externa. But all kinds of blood vessels absolutely have to have a tunica intima. So if you first look at the charts on the right, you can see a comparison between the 5 different kinds of blood vessels: arteries, arterioles, capillaries, venules, and veins. So you can actually see a breakdown as to their composition. We're not using exactly the same terminology in this diagram, but essentially the endothelium refers to the tunica intima, and you can appreciate that every kind of blood vessel at minimum has that. The fibrous tissue on the far right refers to the tunica externa. You can see the arterioles and capillaries do not have that. Smooth muscle obviously refers to the tunica media. Arteries, arterioles, and veins have that smooth muscle, but venules and capillaries do not. Therefore, venules and capillaries are not capable of being vasoconstricted or vasodilated. And we will discuss these sorts of concepts later on, and we will find out that the best way to achieve regulation of blood flow into a tissue by using vasoconstriction and vasodilation is to go after the arterioles. A couple of other noteworthy pieces of information you can see here: the diameter and thickness of the wall of each kind of blood vessel. It shouldn't be a surprise that arteries and veins are the largest of the blood vessels in terms of their mean diameter, with veins being on average the largest. And then as you get closer to the capillary bed, the diameter of the blood vessel gets narrower and narrower. Capillaries having a mean diameter of 8 microns ends up meaning, as we have discussed before, that red blood cells moving through a capillary bed are going to have to move through one cell at a time. That's how narrow things are in the capillaries. And then if you look at the thickness of the wall, even though the vein has a wider diameter than an artery, the artery ends up having a wall that is twice as thick as the vein. And if you look at the diagrams on the left, you can actually see what this ends up meaning. You can see that the vein ends up having a much thinner wall, and that in part contributes to the vein kind of looking like it's collapsed in on itself. Now, the thickness there is kind of a minor issue. The real reason the vein has kind of looked like it's collapsed in on itself here is because veins carry extremely low pressure blood, so there's not exactly a lot of outward pushing forces to keep the vein fully open. The arteries, on the other hand, not only have a very thick wall, but they are carrying highly pressurized blood, and that prevents the artery from looking like it's collapsed, as you see right there. So if we go ahead and focus a little bit on arteries and arterioles, these are going to contain all three tunics that we've seen before for the most part. So the main issue that we want to make sure we understand here is that because arteries and arterioles both contain tunica media, they are both capable of experiencing vasoconstriction and vasodilation. However, we are going to see, again, as I mentioned, really it's the arterioles that we want to vasoconstrict and vasodilate if we are trying to regulate blood flow into a particular tissue or organ. But ultimately, whether it's arteries or arterioles, veins also have tunica media. But you might want to be thinking about this. What is it that you know about veins that would indicate that vasoconstriction and vasodilation would be kind of pointless there? So be thinking about the idea that the veins are on the other side of the capillary bed, and if we want to achieve vasoconstriction and vasodilation, it's usually because we want to conserve resources like oxygen and other nutrients for the tissues that need them most. So be thinking about why vasoconstricting the veins may not be a good way of doing that. So capillaries, as we have said, are the site of gas exchange and nutrient delivery and waste pickup. So when we're talking about blood delivery to tissues, that is a thing called perfusion. So we want our tissues, namely the most needy tissues like the brain and the heart and the skeletal muscle, to be well perfused with blood. In that way, they receive plenty of oxygen and nutrients. So capillaries are the site of tissue perfusion that exists between arteries and veins. Now, you can actually see several different types of capillaries, as you see here. For the most part, most capillaries in the body are going to be of the fenestrated variety. So you'll recall that the fenestrations that you see here, those little Swiss cheese-like holes that exist in between the capillary endothelial cells, those exist because of a lack of tight junctions existing at the interface between neighboring endothelial cells. So that allows for delivery of large molecules like hormones and other things that cannot otherwise pass through the membranes of these endothelial cells to escape the blood plasma and to get into the interstitial fluid. Continuous capillaries exist for the most part at the blood-brain barrier. These endothelial cells do have tight junctions sealing up the gaps in between the cells, so that produces a barrier that allows only molecules that can pass through the membrane to get from one side to the other. If you need a review on the blood-brain barrier, I suggest you go back to chapter 12. So here you can see a capillary bed, and one issue that we can possibly run into here is that the capillaries, as we said before, are extremely thin and extremely narrow. That makes them highly susceptible to injury. So capillaries can and often do rupture. Now, before you start worrying about a ruptured capillary bed leading to a disruption of blood flow, oftentimes what you will see is a direct connection between the arterioles and the venules that completely circumvents the capillaries. These pathways are called anastomoses. So this allows for the continuation of blood flow even in cases in which capillary beds become temporarily damaged. Okay, now if we move on to the venules and the veins, the walls of the venules and the veins are quite thin when you compare to the arteries and the arterioles, as we said, but they tend to have wider diameters, meaning that they have larger lumens that can contain a much greater volume of blood. Now, ultimately, a big issue with the veins as compared to the arteries is that they carry extremely low pressure blood. And ultimately, the job for the veins is to return blood back to the heart. So that can become an issue in terms of dealing with pressure gradients. You'll recall the major concept we introduced back in chapter 19 is that blood will always move from high pressure to low pressure. Well, that makes for a tough job for the veins because they are already dealing with low pressure blood, and the only way they can continue blood along its path is to make sure it's moving to an even lower pressure destination. Well, when you're dealing with low pressures like this, especially for the veins that are below the level of the heart, what we would call inferior to the heart, talking about the veins that start down on the legs and then go up towards the heart, yeah, you're dealing with blood moving from low pressure to an even lower pressure, but you're also fighting against the force of gravity that would cause that blood to pool down towards the legs. So for that reason, many veins, especially those, again, that exist below the level of the heart, they come equipped with valves that allow blood to move through in only one direction. So that once blood does start to make its way up those veins, up towards the level of the heart, these valves close behind it to make sure that the blood doesn't fall back down to the feet because of the force of gravity. And then similarly, we also have two other mechanisms that ensure that blood is always making its way up from the feet up towards the heart. These are called the respiratory pump and the skeletal muscle pump. So basically what this involves is that a lot of these veins actually go in between muscles in the thoracic cavity that help us breathe, and in between muscles in the legs that help us to walk and do things like that. So that when these muscles contract, it squeezes and temporarily pressurizes the blood to make sure that it can keep going up through the legs and up back towards the right atrium of the heart. So another issue with the veins and the venules is that because the pressure is very low, blood flow tends to be fairly slow here. So as it turns out, a good majority of the total blood supply in the body is going to be in the veins at any given time. So having all these mechanisms that we mentioned above, the valves and the pumps, are absolutely necessary to make sure we are always promoting venous return to the right atrium, because as we're going to see later on, if venous return suffers and we're not doing a good job of returning blood back to the right side of the heart, cardiac output is ultimately going to suffer too, because the end diastolic volume will fall and the stroke volume will fall as well. And here's a nice little chart or table here that just does a direct comparison between arteries and veins, talking about some of the things that we've talked about. And you'll see the arteries and veins tend to be polar opposites when it comes to things like the direction of blood flow, which we've discussed, their general appearance, which we've discussed in terms of the wall thickness and the pressure of the blood they carry. Arteries carry high pressure blood, veins carry low pressure blood. Oxygen concentration, we talked about that. Arteries don't need valves because they are already carrying high pressure blood that can fight the force of gravity. And veins do have to have these valves when these veins exist inferior to the heart. Veins that exist superior to the heart, such as the veins that are returning blood from the brain, they don't need these valves because they are already going with the force of gravity rather than against it. Okay, so that is going to do it for this introductory section for this chapter. I thank you for your attention as always, and I will see you next time, and we will start talking about concepts associated with blood flow and blood circulation. Thank you again, and I will see you next time.